So ultimately, in a capillary bed, we're taking blood and we're putting it very, very close to tissue cells across a very thin wall, right? So you have a great setup for good diffusion. Remember, diffusion occurs best when it occurs over a small space, right? So the capillary wall being very small allows diffusion to occur very rapidly. So materials can move down their concentration gradient either into the blood or out of the blood. So diffusion ends up being the way that most things move in and out of the capillary. Okay? Lipid-soluble substances, so connecting back to our polar-nonpolar discussion early on in the course, right? Lipid-soluble materials can readily pass through cell membranes. Because the endothelial cell that makes up the capillary is so thin, things pass very readily through it. So oxygen, CO2, steroid hormones, lipids, all pass relatively unhindered between blood and tissue across the capillary wall, no receptor required, right? Now remember, when you hear no receptor required, that also means there's no regulation, right? So like oxygen always is moving down its concentration gradient. You can't stop a cell from getting oxygen. Like there's no gate for oxygen, right? It's always going to flow because it's passing right through the membrane. It's not using a transporter. There's no point. There's no place to put regulation. All right. So water-soluble things, they either pass through the intracellular clefts or through channels in the membrane. Except for the kidney, all other capillaries are freely permeable to water. All right? Water in the body is never just water. Okay? So where water moves, solute is going to move with it. Okay? So most of our water-soluble substances are moving through water pathways, either through the intercellular clefts or through the aquaporins. Aquaporins are what we call the water channels that are found on cells, except for one little area in the kidney, in the distal collecting system, but we'll talk more about that later. So this exchange is very effective. So water moves through the clefts 80 times faster than it moves through the vessel. Okay, so you have sort of slow moving blood through the vessel, but the fluid is very quickly going in and out between those. So it means water is exchanged between the plasma and the ECF 80 times in the same time it takes the blood to pass through the capillary. Short version, easy to move things that are water soluble in the capillary, okay? To such an extent that by the time you reach the end of the capillary, the material in the blood is in relative balance with the materials in the tissue that it's passing through, right? Remember, if you take a material and pass it through another, diffusion is going to correct the concentration gradients until they're both the same, right? So that ends up being, by the time blood leaves the capillary, its makeup is very similar to the tissue surrounding that capillary. Now, the pores that this water flows through and these materials flow through, like I said in the last slide, they vary according to where in the body you are. So like in the brain, the pores that through which things pass are very, very small, very restrictive. In the liver, they're great big, very free, and in the kidney, they're a specific size. The holes that water passes through are smaller than a protein, okay, but bigger than an ion or an inorganic, right? So in other words, like sodium can pass very quickly in the kidney, but a protein like hemoglobin can't. It's too big, can't get through the hole, okay? The kidney basically works by filtration, all right? So we're going to talk in great detail about what can pass and what can't when we get to that section. Okay, so arterial end, venous end, here's our capillary, so blood's going through the capillary, and then all along that path, we're getting the exchange of water-soluble molecules across that wall. That exchange is between plasma and extracellular fluid, right? So it means that by the time we get to the end of the capillary, the blood leaving has very similar chemical makeup to the ECF that it passed through. Now, the other piece down here is the lymphatic capillary, which we'll talk about at the end of today. We actually have a second set of very thin vessels that collect fluid that doesn't make it into the blood. So we call those the lymphatics, and you can see here that fluid is freely exchanged between the extracellular fluid and the lymphatics all along that path. But we'll talk more about that when we get to the lymph stuff at the end. All right. So the environment of the capillary beds, so we're talking about the interstitium and interstitial fluid. Interstitial fluid is just a specific way of defining extracellular fluid. Interstitial fluid is the extracellular fluid that is outside of the vessels. Okay, so interstitium is the area between the cells where tissues are formed. And, typically when we see sort of pictures and diagrams, that space looks empty or just filled with fluid. In reality, the interstitium does have a structure, right? We have, so we have cells, okay, and then we have our sort of bundles of collagen that give the tissue its shape. And then we have lots of little filaments, extracellular filaments of proteoglycan. That term means that it's part protein, part carbohydrate. All right, proteoglycan. And proteoglycan filaments tend to hold water next to them. They're attractive to water. So by having collagen to give it strength and proteoglycan filaments to hold the water, we have both structure and plenty of fluid for the tissues to get their materials, get their raw materials, and to get rid of their waste products. So together, the collagen and the proteoglycan, the interstitial fluid, it isn't really a fluid at all. It's kind of like a gel, right? In other words, if you dumped it on the table, it would keep its shape, kind of like Jell-O would, right? So it isn't free fluid necessarily. So the gel, the Jell-O, for lack of a better term, it does resist the bulk flow of fluid, but it allows diffusion to occur very readily. So we don't get milliliters of fluid moving, okay? Instead, we get the molecules dissolved in the fluid moving instead, all right, down their concentration gradient. So there's this sort of gel, and then the gel does sort of form little pockets of free fluid called rivulets. And as extracellular fluid, interstitial fluid, increases, the rivulets get bigger relative to the whole ground glass, or the whole interstitial gel, right? So the more ECF we have, the more flow of actual fluid we get, in addition to the flow of molecules. All right, what I'm getting at here is in a typical capillary bed, there isn't a lot of flow of fluid, even though there's a lot of movement of molecules, okay, because of this sort of gel-like structure. So less than 1% of the fluid that's found in tissues flows like a river, right? Instead, it diffuses like the smell in the room, right? Doesn't flow, it sort of moves. Well,